they find more than 40 human skeletons, some of which are abnormally large.
In 1911, giant bones, large human skulls, and skeletons that measured between 7 and 8 feet in height.
For most of the 20th century, American archaeology held a single idea. The first people in the Americas were the Clovis toolmakers. It began in 1932 near Clovis, New Mexico, with fluted stone points found with mammoth bones. Later, radiocarbon dating placed them at about 13,000 years old. Textbooks described migration from Siberia via the Bering land bridge and ice-free corridor. But recently, a 14,000-year-old Alaskan skeleton was sequenced, and just like that, the theory collapsed. Collapse of the Clovis first model. The first hiccup in the Clovis first model came from South America. In the dry valleys of southern Chile, at a site called Monte Verde, excavations led by Tom Dillahay in the 1970s and 80s uncovered something unexpected: wooden structures, stone tools, and preserved plant remains lying beneath peat-rich sediments. Initial claims of extreme antiquity were met with skepticism so strong that many researchers dismissed the site outright. But as excavation methods improved and additional dating was conducted through the 1990s, the evidence held. By 1997, Monteverde was widely accepted as a human occupation site dating to roughly 14,500 years ago, at least 1,000 years before Clovis. So, if humans were already in southern Chile before Clovis appeared in North America, the first arrival at 13,000 years ago model could not survive intact. But this was not the only problem. While Monte Verde was forcing a re-evaluation at the southern edge of the hemisphere, North America was quietly producing its own challenges. In Pennsylvania, at Meadowcroft Rock Shelter, archaeologist James Adavasio and his team uncovered deeply buried cultural layers containing stone tools and plant remains. Radiocarbon dates suggested human presence potentially as early as 16,000 years ago. Repeated analyses and continued excavation strengthened the case that humans were present in eastern North America well before the Clovis culture ever appeared. Meadowcroft did not deliver a single dramatic overturning moment. Instead, it chipped away at the foundation, layer by layer, until the supposed boundary of first occupation began to blur. Farther west, a different kind of evidence emerged from Texas at the Buttermilk Creek complex, part of the Deborah L. Friedkin site. Here, archaeologists uncovered stone tools lying beneath Clovis deposits, sealed in sediments dated to approximately 15,500 years ago. Unlike earlier contested sites, Buttermilk Creek benefited from modern excavation methods and improved dating techniques, including optically stimulated luminescence. The artifacts were simple compared to Clovis points, but their position beneath unmistakable Clovis layers created a clear chronological inversion. Clovis was no longer at the bottom of the timeline, but this gave way to more complications. Ancient DNA and the single origin with structure model. For decades, archaeologists argued over stone tools and migration routes, but it was ancient DNA that finally forced the narrative into something far less cinematic and far more precise. Not multiple unrelated origins, not scattered global founders, but a single ancestral population that carried within itself more complexity than anyone had expected. The turning point came from genetic work beginning in the early 2010s, when sequencing techniques improved enough to recover genomes from Ice Age remains in North America. One of the most important early breakthroughs came from the Upward Sun River site in Alaska's Tanana Valley. In 2013, archaeologists uncovered the remains of 2 infants buried in a hearth-like feature. One genome, labeled USR1, was successfully sequenced in 2018 by a team led by Eske Willerslev at the University of Copenhagen. It revealed a population that had already split from other Native American ancestors tens of thousands of years earlier, yet still belonged within the same broader ancestral system. That system is now often described as a Beringian population complex. It was not a static group standing at a literal land bridge waiting to cross. Instead, it was a dynamic population structure spread across what was once exposed land between Siberia and Alaska. This was, of course, during the last glacial maximum. This region, Beringia, was not just a corridor, but a vast ecological zone, stretching from the Lena River basin in Siberia to the Yukon in Canada, when sea levels were significantly lower, exposing up to 1,000 kilometers of additional land. Genetic evidence places the formation of the ancestral Native American lineage somewhere between roughly 25,000 and 20,000 years ago. Around this time, populations derived from East Asians became isolated in Beringia. That isolation is critical. It was not a short stopover. It likely lasted several thousand years. During this period, the population began to split internally into distinct branches, a process visible in modern and ancient genomes. USR1 from the ancient Beringian lineage is the clearest example of this internal structure. It represents a branch that diverged early from the ancestors of most Native Americans before the major expansion into the rest of the Americas. This means that by the time humans began moving south of the ice sheets, they were not genetically identical migrants moving as a single wave. They were already structured into related but distinct lineages. That insight fundamentally changed how researchers interpret the first Americans. Instead of imagining a small group crossing a land bridge and rapidly populating two continents, the genetic data supports a broader founding population with deep internal diversity. In practical terms, this means that the people who entered the Americas were not a uniform founding tribe, but a set of related populations already separated by time, geography, and partial isolation within Beringia itself. One of the strongest pieces of evidence for this model comes from genome-wide studies comparing ancient and modern indigenous populations across North and South America. Despite vast geographical separation, from the Arctic to Patagonia, Native American populations share a strong genetic continuity traceable to a single ancestral source. This source does not resemble multiple independent migrations from different continents or unrelated population inputs. Instead, all known indigenous American lineages converge back to a shared ancestral node in Northeast Asia, or Beringia. However, single origin does not mean single event. After the initial split from East Asian populations, the ancestral group underwent internal diversification before expanding into the Americas. This is why modern indigenous populations can be grouped into major branches: Northern Native Americans, Southern Native Americans, and others. One of the most influential large-scale genomic studies supporting this framework was published in Nature in 2025, analyzing over 100 indigenous genomes from North and South America. It identified at least 3 deep ancestral lineages contributing to early South American populations, all tracing back to a shared ancestral source. This is where older migration theories begin to break down. Earlier models often imagined multiple waves from different geographic sources: Siberia, coastal Asia, even speculative trans-Pacific arrivals. But ancient DNA does not support that level of external diversity. The Beringian standstill hypothesis fits into this framework as a key explanatory phase. Let's dig a bit deeper into that, shall we? Beringian standstill hypothesis. Genetic divergence estimates consistently place the split between ancestral Native Americans and East Asian populations somewhere around 20,000 to 25,000 years ago. That period aligns with the peak of the last glacial maximum, when massive ice sheets locked up North America and sea levels dropped more than 100 meters. What emerged between Siberia and Alaska was not an open highway for migration, but a vast exposed landmass known as Beringia, stretching from the Kolyma River region in Russia to the Yukon in Canada. It was cold, dry, and steppe-like, but not uninhabitable, and according to the genetic data, it became a long-term refuge. The standstill model proposes that after separating from East Asian populations, a founding ancestral group did not immediately move into the Americas. Instead, they remained in Beringia for several thousand years, potentially from roughly 25,000 to 15,000 years ago. During this time, the population was effectively cut off in 3 directions. Ice sheets blocked the south. Glaciers and Arctic conditions limited westward expansion. The Bering Strait region was a broad but environmentally constrained zone. This isolation is embedded in the genetic clock. All indigenous American populations share a bottleneck signature consistent with a prolonged period of reduced effective population size, followed by rapid expansion. Inside this frozen stage of geography and time, the ancestral population did not remain uniform. Genetic studies, including analyses led by researchers such as Eske Willerslev and colleagues in Copenhagen, show that even before the main expansion into the Americas, the population had already begun to split into distinct lineages. The most striking example is the ancient Beringian branch, represented by the genome of an infant from the Upward Sun River site, or USRR1, dated to about 11,500 years ago, but genetically tracing back much deeper. This lineage diverged early from the ancestors of most other Native Americans, suggesting that Beringia was not a single homogeneous population, but a structured landscape of human groups separated by distance, ecology, and time. That internal structure is one of the strongest pieces of evidence supporting the standstill hypothesis. If the ancestral population had simply passed through Beringia quickly, genetic variation would be shallow and uniform. Instead, what appears is a branching tree, one trunk splitting into multiple limbs before any major migration southward occurred. This implies that Beringia was occupied long enough for genetic drift and substructure to develop, likely thousands of years rather than a brief stopover. In practical terms, the standstill was a phase of evolutionary divergence. Climate data strengthens this interpretation. Between roughly 26,000 and 19,000 years ago, North America was dominated by the Laurentide and Cordilleran ice sheets, which merged in places and formed a continuous barrier across much of present-day Canada. This blocked inland migration routes entirely. Even when partial corridors opened later, they were unstable and ecologically sparse. Coastal routes along the Pacific may have been available earlier, but they still do not fully explain the timing gap between divergence from Asia and expansion into the Americas. The standstill model resolves this mismatch by placing the population in Beringia itself during the glacial maximum, rather than immediately on the move. However, the hypothesis is not without debate, particularly regarding duration and internal movement. Some researchers argue that the standstill may not have been a static isolation zone, but rather a dynamic region with intermittent gene flow. Micromigrations into and within Beringia are a key uncertainty. For example, populations could have shifted seasonally between western and eastern Beringia, or experienced contact with neighboring Siberian groups during warmer phases. Genetic data does not fully exclude these possibilities. Another unresolved question concerns exactly when the standstill ended. Most models suggest that expansion into the Americas began around 17,000 to 15,000 years ago, coinciding with the gradual retreat of ice sheets and the opening of viable ecological corridors. But even this timing is not absolute. Some archaeological evidence, including early sites in South America, such as Monte Verde, suggests that movement into the hemisphere may have occurred quickly once conditions allowed, or possibly even earlier via coastal routes. This compresses the transition from standstill to expansion into a relatively short window. The key insight from genetics is that all modern indigenous American populations, whether in the Arctic, the Amazon, or Patagonia, share a common ancestral bottleneck consistent with this Beringian phase. That means the standstill is not tied to one group or region. It is written across the entire continent's population history. Even highly divergent groups in South America trace their ancestry back through the same ancestral node. This is why the hypothesis has become central. It explains both unity and diversity. Unity comes from a shared source population. Diversity comes from an internal structure formed during isolation. What the standstill model does not support is the idea of multiple unrelated founding migrations into the Americas. There is no genetic evidence for independent continental origins arriving separately from East Asia, Australasia, or Europe during the Ice Age. Signals sometimes interpreted as exotic contributions, such as the faint Australasian affinity detected in some Amazonian groups, are now generally explained as either deep ancestral structure within Asia or statistical echoes of ancient population mixing predating the Beringian isolation phase. They do not require separate transoceanic migrations. The hypothesis also reframes how researchers interpret archaeological gaps. If humans were isolated in Beringia, for thousands of years, then the absence of earlier widespread sites in the Americas is not evidence of absence. It reflects geography. Much of the land bridge that once formed Beringia is now submerged beneath the Bering Strait, meaning large portions of the standstill population's landscape are physically inaccessible today. What survives in Alaska, Yukon, and parts of Siberia is only the edge of a once much larger inhabited zone. Ultimately, the Beringian standstill hypothesis is not a single claim, but a convergence point between genetics, paleoclimate, and archaeology. But the uncertainties remain. Now, we'll address them. Multiple early migrations versus single expansion pulse. When ancient DNA began to be recovered from early human remains across the Americas, starting in the early 2010s, the expectation was simple. If there had been one founding migration, genomes should show a relatively uniform branching pattern. Instead, researchers found something more layered. One of the most important early genomes came from the Anzac child in Montana, associated with Clovis tools, and dated to about 12,600 years ago. Sequenced in 2014, it showed clear ancestry to modernist peoples of the Americas, confirming continuity. But it also hinted that Clovis was already part of a population that had diversified earlier than previously assumed. Then came Kennewick Man, found in Washington state, and dated to roughly 9,000 years ago. Sequencing, completed in 2015, confirmed he was closely related to present-day Native American groups. Clearly, early Americans were not isolated experiments of migration. They were part of long, connected population histories already established across regions. As more genomes accumulated, a pattern began to emerge that did not fit a single-wave model. Instead of one clean branching event, researchers observed at least two deep ancestral lineages within Native American populations very early in their history. One branch is associated with northern Native Americans, the other with southern Native Americans. Genetic studies suggest that this split occurred very early, possibly soon after entry into the Americas or even within Beringia itself. This means that by the time populations were spreading across the continent, they were already internally differentiated. This early north-south split is one of the strongest arguments against a single uniform expansion pulse. If all populations had moved south together in a single rapid migration, genetic differences between distant regions would have accumulated gradually and relatively evenly over time. Instead, what is observed is an early bifurcation followed by long-term regional continuity. Southern Native American populations, for example, show deep ancestry that diverges early from northern groups, indicating that separation happened quickly after or during the initial expansion phase. Archaeological evidence aligns with this genetic structure. Sites across North and South America do not form a simple chronological cascade of occupation. Monteverde in Chile, dated to at least 14,500 years ago, appears almost contemporaneous with or earlier than many northern sites traditionally associated with early human presence. Meadowcroft in Pennsylvania may extend human occupation back to 16,000 years. Buttermilk Creek in Texas adds further evidence of pre-Clovis occupation around 15,500 years ago. These sites are not neatly arranged along a single southward migration route. They suggest rapid dispersal and regional occupation patterns that do not follow a single linear expansion. This is where the idea of multiple pulses enters the discussion. Instead of one migration wave, some researchers now propose that human expansion into the Americas may have occurred in stages. The first pulse could have involved a relatively small founding population entering and spreading rapidly across available landscapes. A second phase may have followed as climate conditions improved, ice sheets retreated, and ecological corridors opened more fully. Genetic evidence does not require multiple independent origins to explain this pattern. All modern indigenous populations still trace back to a single ancestral source population rooted in Northeast Asia and Beringia. However, within that source, structure matters. The ancestral population was not homogeneous at the moment of expansion. It already contained subgroups that had diverged during the Beringian period, meaning that when expansion occurred, it carried internal diversity outward into the Americas. This helps explain why early differentiation between northern and southern lineages appears so quickly after initial settlement. Rather than evolving in isolation after migration, these lineages were already partially separated before or during entry into the continent. Some older hypotheses suggested entirely separate migrations from different continents. Ideas involving Australasian, European, or even multiple independent global sources. These are not supported by current genetic data. While faint and debated signals, such as the so-called population Y, have been observed in certain South American groups, these do not require independent founding migrations. Instead, they are better explained by deep shared ancestry in Asia or complex population structure in the source region before migration into the Americas. No genomic evidence supports separate founding populations entering from unrelated continental origins. The key distinction in modern research is between single origin and single wave. A single origin is strongly supported. All indigenous peoples of the Americas trace back to a shared ancestral population. A single wave, however, is increasingly unlikely. The timing of archaeological sites, the structure of ancient genomes, and the rapid emergence of regional diversity all point toward a more dynamic process. One influential synthesis model now describes the expansion as a structured dispersal. In this view, a genetically diverse ancestral population in Beringia began expanding southward in response to environmental changes around 17,000 to 15,000 years ago. As groups moved, they split, adapted, and diversified as they entered new ecological zones. This produced early regional lineages that can still be detected in modern genetic data. The story is no longer about how many groups arrived, but about how quickly one population became many once it entered a vast new world. White sands and very early human presence. Human footprints pressed into wet sediment, sealed for more than 20 millennia beneath layers of time in White Sands National Park, New Mexico, revealed a whole new angle to the whole story. What makes them unsettling is not just their preservation, but their timing. If the dates hold, they place humans in North America during the last glacial maximum, when massive ice sheets locked most of the continent under frozen pressure, and earlier migration models said no one should have been there yet. The site itself sits along what was once Lake Otero, a vast Ice Age lake that no longer exists. In 2021, a research team led by Matthew Bennett published evidence in Science describing a series of human and animal footprints embedded in gypsum-rich sediments. These prints were not isolated impressions but trackways, multiple steps preserved in sequence, showing direction, stride length, and interaction between individuals. There are also prints of children walking alongside adults. The dating was based on radiocarbon analysis of ancient seeds from Rupia aquatic plants found in the same sediment layers. Those seeds consistently returned ages between roughly 21,000 and 23,000 years before present. If correct, the implications are immediate and disruptive. The conventional timeline places the main expansion of humans into North America after about 17,000 to 15,000 years ago. Following the retreat of ice sheets, White Sands pushes human presence several thousand years earlier, into a period when the Laurentide and Cordilleran ice sheets still formed a near-continuous barrier across much of northern North America. That means humans were present under extreme glacial conditions, in landscapes previously considered inaccessible. The footprint layers themselves were identified through ground-penetrating radar surveys and careful excavation of sediment strata. Researchers found multiple levels of prints at different depths. This suggests repeated use of the lake margin over time. Some trackways show individuals moving at walking speeds of approximately 1.2 to 1.5 meters per second, while others suggest running or hurried movement. One set of prints appears to show a child's footsteps partially overlapping those of an adult, implying group movement rather than isolated passage. What makes the White Sands evidence particularly provocative is that it is not an artifact-based site. Unlike stone tools, which can be redeposited or reworked, footprints are instantaneous records of presence. They cannot be manufactured or transported. If the stratigraphy and dating are correct, they represent direct, unmediated evidence that humans were physically present in the interior of North America during the peak of the last ice age. But the site's dating methodology has not gone unchallenged. The primary radiocarbon dates come from aquatic plant material which can sometimes incorporate old carbon from dissolved sources in lake water. This reservoir effect can potentially make samples appear older than their actual age. Critics have argued that this could artificially push the footprint dates deeper into the past. To address this concern, a follow-up study published in 2023 by researchers, including Kathleen Springer and others, applied additional stratigraphic and sedimentological analyses. They reinforced the original conclusions. They examined multiple independent layers, including pollen assemblages and sediment deposition patterns, which supported the idea that the dated layers were contemporaneous with the footprints rather than contaminated or mixed. Even with these reinforcements, parts of the archaeological community remain cautious. The debate is whether the absolute dating is precise enough to anchor them at 21,000 to 23,000 years. If the earlier dates are correct, the White Sands evidence forces a rethinking of migration routes. The traditional ice-free corridor hypothesis cannot easily accommodate human presence at this time, because the corridor between the Laurentide and Cordilleran ice sheets is believed to have been ecologically viable only after roughly 15,000 years ago. That leaves two alternatives. Either humans moved along a coastal route earlier than previously assumed, or they entered and dispersed into interior regions during brief climatic windows when glacial conditions allowed localized passage. The coastal migration model gains renewed relevance under this scenario. Along the Pacific Rim, exposed continental shelves during the Ice Age could have formed a kelp highway, a productive marine ecosystem stretching from Northeast Asia through Beringia and down the western coast of the Americas. This route would have provided marine resources, coastal refuges, and relatively continuous movement corridors even during glacial maxima. Another possibility is that White Sands reflects the early inland penetration by populations that had already been on the continent for some time. Humans may have entered earlier via coastal or northern routes and subsequently spread into interior basins, like the one that once held Lake Otero. That would mean the White Sands footprints capture a moment of established presence, not initial entry. Genetic data does not contradict either scenario outright, but it does complicate them. Ancient DNA suggests a primary expansion into the Americas occurred after roughly 17,000 to 15,000 years ago from a Beringian source population. If humans were present in New Mexico 5,000 years earlier, it implies either earlier, as yet unsampled population movements, or a need to reconsider the timing of dispersal from Beringia. Some researchers propose that early exploratory migrations may have preceded the main expansion pulse, but left little or no genetic signature in modern populations. But there's more. The 14,000-year-old Alaska skeleton that changed the migration story. For decades, the peopling of the Americas was taught as a clean, linear event. One migration, one founding population, one movement across the Bering land bridge around 13,000 years ago, followed by rapid expansion southward through an ice-free corridor. That model survived for most of the 20th century because the evidence seemed to support it. One of the most important pieces of that puzzle came from a remote cave system on Alaska's Seward Peninsula, Trail Creek Caves. The human remains recovered there had actually been excavated in the late 1940s by Danish archaeologist Helge Larsen, alongside stone tools, animal bones, and Ice Age hunting debris. For decades, the remains sat in museum collections with limited analytical value because the technology to extract ancient DNA simply did not exist. Only after major advances in genomic sequencing during the 2010s did researchers begin to understand what those bones were actually preserving. When genetic material from ancient Alaskan remains, including material connected to Trail Creek cave studies and the broader Beringian DNA work, was compared with genomes from later Native American populations, scientists realized something fundamental. The first peoples entering the Americas were not genetically uniform. They were already divided into distinct lineages before large-scale expansion southward even began. This realization exploded out of the broader Alaska ancient DNA revolution led by researchers, including Ben Potter and Eski Willerslev. In 2018, sequencing of the Upward Sun River infants from interior Alaska identified a previously unknown branch now called the ancient Beringians. These individuals belonged to a lineage that split from the ancestors of other Native Americans roughly 20,000 years ago, during the height of the last glacial maximum. That discovery changed the migration debate immediately. Under the old Clovis first framework, the Americas were supposedly populated by a single homogeneous group crossing into Alaska around 13,000 years ago. But the Alaska genomes showed that by the time populations dispersed into the Americas, they had already undergone internal diversification for thousands of years inside or near Beringia itself. The Trail Creek Caves material became important because it supported this emerging picture of a structured Beringian population system. Beringia was no longer viewed as a temporary bridge that people hurried across. It increasingly looked like a long-term refugium. Researchers no longer saw a single migration wave, but branching groups with distinct ancestry before spreading across the Americas. This also reshaped views on population Y. Earlier theories suggested direct Australasian migrations, but new Alaska DNA evidence instead pointed to ancient population diversity already existing within Eurasia and Beringia. Ice Age Northeast Asia was not a single uniform population, but a fragmented mosaic of groups separated by glaciers, geography, and climate barriers. Within this structured landscape, the ancestors of Native Americans formed, carrying inherited layers of older Eurasian ancestry that later became unevenly distributed across the Americas. In this context, population Y is no longer seen as evidence of an unknown migration from Australasia into South America. Instead, it is better understood as a faint genetic echo of deep population structure already present within the ancestral Native American gene pool before expansion into the Americas. Ancient DNA from individuals like Kennewick Man reinforced this picture. Sequenced in 2015, his genome showed clear genetic continuity with modern indigenous populations of the Pacific Northwest, rejecting earlier speculation that early North Americans were unrelated to present-day Native Americans or derived from separate global migrations. At the same time, archaeology pushed timelines back further. Monteverde in Chile, White Sands footprints. They both challenged the Clovis first model. Today, we know that humans were present in the Americas earlier than previously assumed. Well, that's it for now. Thanks for watching. Did you like this video? If you did, please consider liking, dropping a comment, and hitting the subscribe button so that you don't miss out on our new uploads. Also, don't miss this video you see on your screen right now. It's truly unbelievable.